Hi, I am an enzyme involved in degradation of proteins. You can call me Protease. I am also known as proteinase or proteolytic enzyme. I perform complete degradation of proteins to free amino acids, which is termed as proteolysis. Proteolysis occurs not only in digestive tract but also in every cell, that is, in lysosomes, in cytoplasm, and other parts of the cell. Actually, proteolysis is a ubiquitous mechanism that the cell employs to regulate function and fate of proteins. It requires series of enzymes with different specificity. Generally, specificity is the ability of an enzyme to select a specific substrate from a range of chemically similar compounds. Since the enzyme and the substrate exhibit complementary structural and conformational properties, specificity is a molecular identification process. Because of its complex structures and high molecular weight of proteins, enzymic proteolysis is extremely complicated. Most proteases are quite specific with regard to which peptide linkages they can split. I am found in plants, animals, and in microorganisms too, and some foods like kiwi fruit, ginger, asparagus, yogurt, etc. And I am taken in the form of supplements also. In plants, I am found as papain, bromelain, and in animals, I am mainly produced in stomach and pancreas and found as trypsin, pepsin. And in microorganisms, subtilsin and thermolysin are my examples. On the basis of reaction I catalyze, I am classified into 6 distinct classes: aspartic proteases, glutamic proteases, and metalloproteases, cysteine proteases, serine proteases, and threonine proteases. Although glutamic proteases have not been found in mammals so far. And on the basis of pH, I am classified as acidic proteases, neutral proteases, and alkaline proteases. And on the basis of mechanism of catalysis, I am classified as aspargine peptidase, aspartic peptidase, cysteine peptidase, glutamic peptidase, metallopeptidase, serine peptidase, and threonine peptidase. And finally, on the basis of the site of enzyme action, I am classified as exopeptidases and endopeptidases. Endopeptidases and exopeptidases. Exopeptidases catalyzes the removal of an amino acid at the end of the polypeptide chain and releases a single amino acid or dipeptide from the peptide chain. Endopeptidases. They catalyze the breaking of a peptide bond within the polypeptide chain and cannot break down peptides into monomers. Exopeptidases are classified into types. Firstly, aminopeptidases. They cleave off a single amino acid from the amino terminus. And secondly, dipeptidyl dipeptidases. They cleave off dipeptide from the amino terminus. And carboxypeptidases. They cleave off a single amino acid from the carboxy terminus. And finally, dipeptidases. They hydrolyze dipeptides into 2 single amino acids. And endopeptidases are also classified into types: trypsin, chymotrypsin, elastase, thermolysin, pepsin, and endopeptidases V8 are some of their types. I am widely known for my role in digestion and catabolism of proteins, blood coagulation, cell division, growth, and migration. Protein recycling and transport across membranes mediate immune response, wound healing, and muscle soreness. And I am also involved in mediate processes that are frequently irreversible, like maturation of cytokines and prohormones, breakdown of intracellular proteins, regulate fate mechanism, localization and activity of many proteins, modulate protein-protein interactions, and create new bioactive molecules that contribute to the processing of cellular information, remodeling, heat shock, and unfolded protein responses. These are my well-known functions. Generally, polypeptides can be cleaved either chemically or enzymatically. Enzymes that catalyze the hydrolytic cleavage of peptide bonds are called as proteases. Proteases fall into four main mechanistic classes like serine, cysteine, aspartyl, and metalloproteases. In the active sites of serine and cysteine proteases, the eponymous residue is usually paired with a proton withdrawing group to promote nucleophilic attack on the peptide bond. Aspartyl proteases and metalloproteases activate a water molecule to serve as the nucleophile, rather than using a functional group of the enzyme itself. However, the overall process of peptide bond scission is essentially the same for all protease classes. Some of my examples with their respective function are trypsin, which is found in pancreatic juice and break the proteins, peptones and proteases. And some more examples like chymotrypsin, carboxypeptidases, elastases, nucleases, collagenase, dipeptidase, pepsin, renin, thrombin, plasmin, renin, insulinase, chymases, tryptases, and neuralases. My top benefits includes my ability to allow for the digestion of proteins and the absorption of amino acids, boost immune function, promote cardiovascular health, accelerate tissue repair, and possibly prevent colon cancer. When I am taken as a supplement, I may cause side effects like allergic reactions, and another side effect that has been reported is stomach upset, including diarrhea, nausea, and vomiting. I am having a wide range of applications in different industries includes meat processing industries, baking industries, dairy industries, leather industries, detergent and pharmaceuticals. I'd like to stop here if that's all right. And do remember my name, Protease.